Good morning, good afternoon, and thank you very much for listening to my presentation today. I'm Virginie Goubert, Global Application Specialist at Akoya Biosciences. I'm very happy to present you our solutions for multiplexing and spatial phenotyping in situ and at single-cell resolution. Akoya Biosciences has been the leader in the spatial biology revolution, transforming discovery and clinical research with groundbreaking multi-parameter tissue analysis solution for spatial phenotyping. Our technology are well proven through a large install base worldwide and hundreds of publications. We provide a complete end-to-end solution from reagents to imaging instruments and software analysis. As you can see on this slide, Akoya has evolved very rapidly the past few years, starting with 3 employees in 2017 up to 160 employees nowadays, and partly through the acquisition of the multiplexing tissue imaging division from PerkinElmer in 2018. In 2019, we launched a Codex platform, our ultra-high plexing solution invented by Gary Nolan from Stanford University. This technology was developed mainly to allow any lab to perform super high plexing, so up to 50 markers on a single tissue section with a single cell resolution. And we're very proud to have more than 500 instruments installed worldwide and a growing number of publications, up to 200 in the last 2 years. So what do we call spatial phenotyping? Spatial phenotyping means looking at biomarkers in context, because we believe every single cell counts. And we have developed this new concept to respond to a new biomarker paradigm in tissue analysis. You probably all very well know the power of proteomics and genomics when it comes to looking at tumor mutations and gene expression in order to better understand the tumor progression or self-defense mechanisms. But we also know that we lose the tissue context, and context matters because we need to understand the interactions between those different phenotypes, and we need to look at every cell to better understand their role within the tumor microenvironment. Immunohistochemistry has always been a powerful tool when it comes to studying biopsies, but we're quickly limited in terms of number of plex one can do. And past clinical trials have demonstrated that looking at just 1 or 2 checkpoint expression is not enough to stratify patients and direct immunotherapy. And that's why we do what we do at Akoya. We've seen this gap between genomics, transcriptomics, and standard immunohistochemistry. We understand researchers want a complete solution from discovery to clinical research, with different requirements throughout the process. And that's why we came to further develop 2 different kinds of platforms. Codex as an ultra-high plexing discovery tool, which enables to image a very high number of markers per tissue section. And then our Phenoptics platform, including reagents, multiplexing imaging instruments, and data analysis software for the everyday lab routine in pathology labs or clinical environment. With Phenoptics, we can do a very high number of samples per day, and we can image a 6-plex in less than 10 minutes. And both platforms allow to image every cell across the entire tissue section, which enables researchers to build detailed network maps and analyze spatial interactions, leading to discovery of distinct cellular neighborhoods and architecture within the tumor microenvironment. As I previously said, Codex is our biomarker discovery platform, making spatial biology broadly accessible to any lab as it integrates standard microscope. It has been voted one of the top 10 innovation in 2019 for its ultra-high plexing capability into spatial context. It is an end-to-end walk-away solution with proprietary reagents and software. The protocol is very simple and fully automated, with the software controlling the Codex stainer on the side and the associated microscope. Codex relies on a repetition of staining and imaging cycles with oligoconjugated antibodies revealed 3 by 3 with fluorescent antisense reporters. The only step one has to do is to cocktail antibodies as much as they want, fix them to the tissue, and walk away. The Codex staining platform and microscope does the rest. Codex staining has been applied on different kinds of tissues and samples. We have now many proof of concept and publications and panel developed on melanoma or breast cancer, brain tissue, lung, skin, gut, kidney, spleen, and also diverse sample types such as PBMC extracted from blood, organoids, or even iPSC cultured cells. So this allows us to respond to different biological questions within diverse disease areas. And we have a particular focus on oncology, immunology, infectious disease, neuroscience, and developmental biology. You might think that looking at several biomarkers on a single tissue section is complex, but I will walk through a classical Codex experiment, for example here on breast cancer, where we stained FFPE tissue section of stage 2 ductal adenocarcinoma with a cocktail of 36 antibodies. And you will see that it's very easy to visualize cell population because you have this view of the entire tissue like you will have in standard immunohistochemistry or immunofluorescence. And as you can see here, we see a tissue of 2 millimeter by 3.5 millimeters, so quite a good size. And every antibody, every marker is color-coded with the color you like. And as I'm toggling through those different images, you can see those different markers represented on every image. So I'm not an expert in breast cancer or breast tissue, but I know breast tissue is known to have many different types of epithelial cells. So for these specific breast cancer panels, it's a lot of epithelium markers. Just on this area, we counted about 63,000 cells, and we could see that there is a lot going on. And that's what we do at Akoya. We really look from an unbiased manner the spatial biology and the specific interaction of different populations through an entire tissue section. So after cell segmentation and unbiased clustering, one can start to define and see relevant cell types and interesting distribution and networking of those cells. Codex comes with a multi-analysis viewer or analysis software, which enables to cluster and organize those diverse phenotypes in t-SNE plot. Here you can see that some populations are obviously more prominent than others. The good thing also with our analysis software is that you can really match what you're seeing on the t-SNE plot back to the images. So here we can see this big cluster, cyan cluster of luminal L1 cells represented in cyan on the image, and this very more discrete population of basal cells also surrounding the ducts in here represented in dark blue. And looking a bit more closely into this like basement membrane, we could identify really where luminal cells, which are keratin 8 and keratin 14 positives, and only 44 cells could be counted on this image out of 63,000. So they're very rarely expressed. And to be honest, when we internally developed this breast cancer panel or proof of concept in our R&D, we had no idea what to look at or what to expect. But we got interested by those rare cells and started to look at publications, and we found a publication recognizing that those keratin 8 and keratin 14 positive cells could be potentially cancer cells of origin for specific breast cancer subtypes. So the take-home message here is that a cell needs to be seen in order to be discovered. And every cell matters and context matters. Even rare cells or underappreciated cells could be shown to be biologically and even clinically relevant. And that's the beauty of Codex, this unbiased discovery capability at a single cell level. And Codex technology could be complementary to transcriptomics or genomics data, where RNA or gene expression are usually quantified on ROI, on region of interest, grouping cells together, which is more some kind of spatial approximations. But with Codex technology, we stain and image every cell, so we have a truly spatial analysis of phenotypes. And where it becomes really interesting and valuable is when we map those different cells and we look at interactions between them, networking, and we do some kind of population neighboring analysis. So last but not least, I would like to present also the data analysis software, which comes with the Codex machine. So we call it MAV, Multi-Analysis Viewer. And it's based on Fiji, and you can analyze your data in many different manners depending on the biological questions you have and the answers you want. So first, what you do is visualize your, you can visualize your tissue by toggling on and off your markers of interest, like I've shown you previously on breast cancer. And after cell segmentation, you can find or define your cell population in several ways. For example, you can manually gate a cell population either by histograms or scatter plot representation based on their expression level. Or you can use a t-SNE plot, like I've also shown before, where you can see the abundance and distribution of cells. And you can also take distance between cells into consideration and generate some kind of circus plot. All is possible within the MAV, but you can also use third-party software if you want to push the analysis even further. And to go a step further, Gary Nolan, the founder of Codex Technology, and his team at Stanford University published last year this very exciting study where they applied 56 biomarkers, 56 antibodies on 140 tissue samples of 35 colorectal cancer patients. And they applied this cellular neighborhood analysis, kind of like a CT mapping algorithm, where they could reveal a very structured spatial organization of the tumor microenvironment. And so with Codex, they were able to demonstrate an altered organization of the tumor and immune components in low versus high risk patients. And they could also see a local enrichment of PD-1 positive, CD4 positive T cells correlating with survival in high-risk patients. So I really encourage you to read this publication, which is really fascinating. Akoya is also very excited to support the Human Cell Atlas project, and we have collaborations worldwide. For example, with Dr. Kai Kessenbrock from University of California, who recently did a webinar with us presenting his work with Codex, and SciLife in Stockholm, where Dr. Charlotte Stadler and Emma Lundberg from the Human Protein Atlas, working on the Human Protein Atlas project, have had a Codex system for quite a while. And quoting Charlotte, highly multiplex methods represent the next step in the field of spatial proteomics and immunohistochemistry, which we strongly believe at Akoya. So circling back to my example on breast cancer study, once those cell population or relevant biomarkers are discovered thanks to the Codex platform and ultra-highplexing, then we can start to think, what is it really relevant, and how can I test it on a much larger group of samples or clinical cohort? And that's where the Phenoptics platform comes into play. As I showed you at the beginning of my presentation, we have a solution for everyone through the translational research continuum. So Codex as a discovery tool, which I showed you in the first part of this presentation. And for higher throughput and clinical settings, we have the Vectra Polaris. Our Vectra Polaris has been built from the ground up to fit clinical needs and clinical research applications. We now have the medical product certification, ISO 13485, and the Vectra Polaris is more and more used in clinical trials worldwide. Here again, we provide an end-to-end solution, as our Phenoptics workflow is made of multiplexing staining reagents that we call OPAL, imaging instruments in order to perform in a very high throughput up to 8 markers per single tissue section, and a data analysis and storage software platform. So for the staining part, we rely on multiplexing kits that are fully automated on Leica Band or Discovery Ultra from Roche. The imaging scanner called Vectra Polaris can image up to 60 slides per day thanks to a very fast scanning, multispectral scanning mode to perform a 6-plex in less than 10 minutes. And then we have a data analysis software called Inform in order to perform cell segmentation, phenotyping, immunoscoring, and so on. And if one wants to do further data analysis using third-party software, it's also doable, and we have a collaboration with Halo and Visiopharm in order to do whole scan analysis. So first, what is OPAL? OPAL is based on TSA, Tyramide Signal Amplification Reaction. So as it says, amplification means an improvement in sensitivity and dynamic range for a better quantification. But also the beauty of OPAL is that we can multiplex using the same primary antibody species if we'd like. So we don't have to play anymore on different species for the primaries, which is often difficult to find. So we can do today up to 8 markers plus DAPI. And the first thing to do is really just to prepare your slide like you will do for any FFP tissue section with antigen retrieval. And then you perform repetition of staining cycles every time using marking a different epitope with a different OPAL dye. And as many as you would like to include in your assay. OPAL can be fully automated overnight or over 11 hours using the Leica Band RX or also Roche Discovery Ultra. We have kits dedicated for automation, and it brings you really the speed, flexibility, and consistency one needs in a lab routine. In order to speed up your assay development, we also provide kits depending on the number of markers you would like to do, depending if you want to do manual or automation, and depending on the tissue species you would like to do. If you want to do human or mouse tissue, we have a solution for you. As I said, we have now 9 fluorochromes, so you can do 8 markers plus DAPI, and it will bring you really the flexibility you need while we provide all the reagents and buffers to perform your assay. And if you'd like to start right away with already pre-validated, pre-optimized solution, we have now carcinoma and melanoma kits, including all you need, including the primaries and all you need to perform your assay. And we're specifically looking at lymphocytes and FOXP3, so for regulatory T cells. And we include in our kit PD-1 and PD-L1 checkpoints for immunotherapy research. And if you'd like to push your study even further by including 2 more markers to our panel kits, you have the possibility to do so with Vectra Polaris and the multispectral imaging. So those images were shared by University of Surrey, where they have a Vectra Polaris, and they were able to perform this 9-color assay on esophageal adenocarcinoma, and they're pushing the research for bladder and prostate cancer. So OPAL is really the solution to everyone who wants to start multiplexing. Whereas you want flexibility and work with your favorite antibodies and the antibodies you've pre-validated in your lab, we have detection kits available to you. And if you want to start right away with ready-to-use pre-validated panel kits, those panel kits that I've presented are made for you. So Phenoptics is a whole package, starting with OPAL multiplexing reagents, and then moving on to high-throughput multispectral scanner, Vectra Polaris, in order to image those 6 or 8 markers on your slides in a fast manner. And then the last piece of the puzzle is the data analysis part. And for that, we have a machine learning software called Inform, which enables to do cell segmentation, tissue classification, cell phenotyping, and immunoscoring. We also developed RStudio algorithms in order to look at spatial distribution of cells and do proximity assessment. For the sake of time, I cannot go into too much details about this bioinformatics part, but I would like to let you know that we're developing a secure, integrated, cloud-based platform to go along the Vectra Polaris for an automated, fast data analysis and reporting. On top of performing data analysis through the cloud, this will enable for scientists to collaborate and exchange data via this cross-platform integration. We have nowadays many publications demonstrating a high concordance, a high correlation between DAB standard immunohistochemistry and OPAL multiplex staining. And we're very proud to have made the cover page of several high-impact journals, beyond IO, where we've been the pioneer in multiplexing. Now we can see our multiplexing solution being used in transplantation, developmental biology, infectious disease, and so on. I would also like to let you know that last year we have performed a big multi-site study including different institutes across US, such as Providence in Portland, Providence Health and Services, MD Anderson Cancer Center in Texas, Yale University and Johns Hopkins on the East Coast, along with the pharma BMS. And really the goal of this multi-site study was to assess the analytical performance of phenoptics to support eventually immuno-oncology clinical trials and clinical testing. So each institute has stained a 6-plex panel, which is composed of PD-L1, PD-1, CD8, CD68, FOXP3, and cytokeratin. And we looked at the concordance of staining and imaging across for immune cell densities. And we obtained a very good correlation with the R-square of 0.86, which shows the reliability of phenoptics across different sites. I'm also very happy to share this recent publication in JAMA, Journal of the American Medical Association, which described a meta-study reviewing different modalities and their predictive value across 44 selected published studies and regrouping 10 different cancer types on about 8,000 patients. So this multi-institutional study regrouping Johns Hopkins, Yale Cancer Center, Northwestern University, and Vanderbilt University really showed that multiplex immunofluorescence, which preserves the spatial context of cellular expression and whole tissue architecture, more precisely predicts response to immunotherapy compared to gene expression profiling, tumor mutational burden, or current single marker immunohistochemistry. Lastly, I'm really excited to share this ongoing study, one of a kind, where we're applying astronomy algorithms to spatial phenotyping and tissue analysis. So it's in collaboration with Yanis Taub from Johns Hopkins and the astronomy department, using this very powerful Sci server, which has been made to analyze trillions of miles of galactic space to apply to spatial analysis of tumor tissue. So we're going from stars to cells, and now we expect spatial phenotyping to identify where cell type and predictive biomarkers, starting with melanoma patients, where we already identified cytotoxic T cells expressing FOXP3 and PD-1 as a relevant phenotype, and we would like to expand it to other kinds of cancers. If you want to read more about our technologies and find publications, protocols, videos, and look at upcoming webinars, please visit our website, akoyabio.com. And now I would like to thank you very much for listening to my presentation, and I'm happy to answer any questions you might have.